Okay, can you all see the slides?
Yes.
Okay, very good. So from today we can start with neurons, okay? We are now studying about the central nervous system. And what is the main function of central nervous system? Anyone? What is the main function of central nervous system? Okay, we will look. Okay, again we can look about it. And so here today we can go with neurons, okay? So here, when studying about the neurons, you must know these are excitable cells, okay? These are not like other cells, these are excitable. These cells could able to transmit the electrical impulse along their length, okay? The first thing is neurons is excitable cells and they could able to transmit the electrical impulse. Okay, this transmission of electrical impulse is very important in the central nervous system. And here with that neurons, we are also having some supporting cells. Okay, here the supporting cells are responsible for making the myelin sheath. Okay, when we are studying about the structure of neuron, we can study about the myelin sheath. So these supportive cells are responsible for making the myelin sheath. And also they are providing the nutrition and some phagocytic roles or phagocytic activities. Okay, so the supportive cells have three main functions. The first one is providing nutrition, supporting the neuron, and they are also having some phagocytic roles. Okay, please, all of you, before studying about this slide, better you write these notes on a paper or straight on your practical book. I will allow the time for it, okay? Please, then taking a screenshot, you can write it. When you're writing, you can get some ideas regarding the points on the slide. So please, I will allocate about 2 minutes. Please write it or at least read the slide. So here these neurons are specialized cells that respond to stimuli and produce impulse, okay? And also it transmits the information to a distant site. So what are the functions it is doing? It is responding to the stimuli. So what is stimuli? Anyone? Any example for the stimuli?
When we feel heat, we suddenly raise up the hand.
Okay, very good. So that is the stimuli. Okay, when somebody calling, we are turning to the sound. No, so the sound is stimuli. So any changes in the internal or external environment can act as stimuli. So here the neurons are the cells that are responding to the stimuli and they are producing an impulse. Okay, impulse means electrical impulse. And they are transmitting that information to a distant site. So here now then we can study about the parts of neurons. The first one is cell body. Okay. So in this picture you can see this is the cell body. Okay. This part is cell body and it is having nucleus, dendrites, and this is the point where the axon starts. So here cell body also can be called as soma. It consists of a large round nucleus surrounded by cytoplasm. And within the cytoplasm we can see some normal cell organelles. Okay, here in this picture, in this picture you can see, wait. Okay, can you see the slides now?
Yes.
Okay. So within the cytoplasm, there are also some normal cell organelles. Okay, normal cell organelles of course will be there inside the cytoplasm. So this is the structure of a typical neuron. Okay, it may come to a mid-examination. Okay, you may be asked to draw the neuron and describes about the parts and write the functions of all the parts. So dendrites originate from the body of neurons, and the neurons are having the dendritic spines on the surface, and dendrites act as the receptive region of the neuron. So the last point is very important. Dendrites are the receptive region of the neuron. So here you can see these are the dendrites. Okay, these portions are the dendrites. So this is the cell body, and these parts are dendrites. So this is just a picture of somatic motor neuron and automatic motor neurons. So this is also a picture of neurons. So these are the dendrites. Here this is the cell body and these regions are axons and the message is conducted in this direction. Okay, in these neurons. So here you can see the synapse. Axon terminal synapses with the dendrites on the target cells. So this is the synapse. Okay. So this is another neuron. So then we can study about the axons. Okay. So based on the parts of neurons, just now we see the cell body and we studied about the dendrites. Cell body is having a central nucleus and it is having almost many cell organelles also, and dendrites are the parts which is receiving the signals from the outside. So here axons, this axon is specialized to allow an impulse to be transmitted along its length. So here along the length of the axons, the impulse is transmitted. And here this nerve fibers, the axons are covered with whitish shed called as myelin. Okay. So here This region is the axon region, and this axon is covered with myelin shed. Okay, can anyone say which kind of cells produce, or what are the cells that are producing the myelin shed? How can we call that cells?
Schwann cells.
Okay. Okay, here this main function of the myelin sheath is to protect and insulate the axon and increase the speed of the conduction. Okay, these two points are very important: protection and insulation of the axon, and increase the speed of conduction of the impulses. So here we can now, from this we are going to study about the supportive cells. So oligodendrocytes are a type of supportive cells. Okay. In the central nervous system, CNS, the collection of myelin fibers are called as white matter, and the collection of cell bodies are called as gray matter. Okay, the white matter is made up of myelin shed, and the gray matter is made up of cell bodies. Okay. Please note this point. You may be asked this question on your spot exam, not spot, on your quiz examinations. Okay. And the collection of cell bodies is called as nuclei. Okay, synapse. Synapse means where the axon terminal meets another cells is called as synapse. Okay. It is consisting three main parts, that is presynaptic membrane and a synaptic cleft and postsynaptic membrane. Okay. I'll show you some videos. On the videos you can clearly understand what is synapse and we can identify these regions. Here, this axon terminals contain the synaptic vesicles, and the synaptic terminals of one neuron can synapse to any part of the adjacent neuron. So here, these terminologies are important. When the neuron synapses on the skeletal muscle cell, it creates a specialized junction called as neuromuscular junction. So, neuromuscular junctions is a synapse between the neurons and skeletal muscles. If the neuron synapses with a gland, it is called as neuroglandular synapse. So how the neuroglandular synapse form? The synapse between the neuron and gland. Okay, there are two types of synapses, electrical and chemical. So in your discussion part, you have to write the variation between, so difference between these two synapses. So these are just pictures regarding the synapses. So this is a picture of chemical synapse, right? And this is the picture of myelin sheds. So the myelin sheds are main function of myelin sheds are insulation and increasing the speed of impulse. Okay, now we can study about the supportive cells. So the neurons are the fundamental cells of nervous system. So these are all responsible for transmitting the signal. Even though they are having the main function of the nervous system, there is only 10%, they make only up to 10% of the nervous tissues. Okay, the remaining 90% of the nervous tissue is having supporting cells, and they can be collectively called as neuroglia or glia. So their main function is supporting, protecting and nourishing the neighboring neurons. Okay, three main functions: supporting the neurons, protecting the neurons and also nourishing the neighboring neurons, both in the central and peripheral nervous system. So here examples of this supportive cells are very important. Astrocytes, oligodendrocytes, Schwann cells, satellite cells, all these are supporting cells. So please again remember the neurons are only 10%. The other 10% is made up of several cells. They can be collectively called as neuroclia. So these supporting cells have the function to support, protect and nourish the neighboring neurons. And examples for the supporting cells are astrocytes, oligodendrocytes and satellite cells. So here you can find the white matter and the gray matter in this spinal cord. Okay, this picture is very important. I think last time for the spot questions, for your seniors had this picture. Okay, so in this picture you must be able to identify the neurons and neuroclear cells. So with that we can stop the nervous system. Okay, I just want you to ask that up to when you are having the online classes? What is the last date for your online class? Or are you having any physical classes after this? Can you all see the screen, the video?
Yes, ma'am.
Okay, wait.
In this video, I'm going to go over the basics of the is the basic unit in the nervous system in a simple animal like.
Can you all hear the sound of the video?
Neurons, but what they allow take information
Yes or no?
from their surroundings and respond to that information. If you look at how those surroundings are connected,
Okay, fine.
very complex. Now humans are quite neuron. The neuron Anderson in it. Hi, it's Paul Anderson, and in this video I'm going to go over the basics of the neuron. The neuron is the basic unit in the nervous system. In a simple animal like a nematode worm, they only have 302 neurons, but what they allow them to do is to take information in from their surroundings and respond to that information. If you look at how those 302 neurons are connected, it's very complex. Now, humans are quite a bit bigger, and we have 100 billion neurons in our body. Most of those are centered in the central nervous system, in the brain, and the spinal cord. But the basic unit of our nervous system is the same. It's the neuron. If we look at the neuron anatomy and remove all the other neurons, the center is going to be what's called the cell body. This is a typical cell. It's a metabolizing cell. It's taking in energy. It's using that energy. And so it's going to have a nucleus. It's going to have Golgi apparatus, mitochondria. It's a typical cell. But there are extensions of those cell bodies called the dendrites. Those dendrites are connected to other neurons, and they're taking information in the form of neurotransmitters. In fact, this whole side of the neuron is receiving information. We then have the axon. The axon is where we're transmitting information. It begins at the axon pillar and is going to move to the terminals. Now remember that the terminals, there's a synapse that connects it to other neurons. But the signal is going to move in this direction. We're taking in information from the dendrites, the cell body, and then we can send information to the next neuron. Now, how do we do that is through an action potential, and I have a video that I'll put a link to where I go into the specifics of how that occurs. But certain axons in our body are really, really long, and for that information to travel quickly, we will wrap them in myelin sheaths. It's a Schwann cell that's wrapped around it. The way it works is we put the voltage-gated channels in between that, and it acts to insulate that nerve. It allows the information to travel more readily down that axon. And we generally say if you have an axon that's wrapped in myelin sheath, we call that a nerve fiber inside our body. Now we're taking in information as well. So there's going to be another neuron that's connected to both the cell body and the dendrites of that cell. But there's going to be a gap between these two, and we call that a synapse. It's a connection between neurons. Again, I have another video on the synapse if you want to dig into the specifics, especially of how chemical synapses work. Neurons are classified in two ways. The first way is due to their structural classification. So if we take the cell body of a typical neuron, like this multipolar neuron, the way you can figure out what type of a neuron is find the cell body and figure out how many poles are coming off of that cell body. So if we count them, we have a number of different dendrites that are coming off this neuron. And then we have one axon that's coming off. And so we call that a multipolar neuron. This is the most common neuron that we have in our body. If we look at the next one, we find the cell body again. How many poles do we have coming off of it? We've got the axon coming in, out, and the dendrite coming in, and so we call this a bipolar neuron. Bipolar neurons are very rare. They transmit information from the receptors in our eye, receptors in our nose. If we look at the next one, here's the cell body. You can see there's only one pole coming off of that. It's actually a fused axon and dendrite, and we call that a unipolar neuron. Now, in vertebrates, it's sometimes referred to as the pseudo-unipolar neuron. These are going to be most of the sensory neurons in our body. They're taking information in and transmitting that back into the central nervous system. And then if we look at the next one, if we look at that, you can see there's no axon on it at all. So we call that an anaxonic neuron. These are found in the brain, and they're not super understood. Now, there are a lot of other structural classifications, but these are the general ones that you're going to find in a typical anatomy class. Now, neurons can also be classified according to their functional classification. Those that take in information, stimulus coming in from our environment, and then the response that we have. So if we kind of move that to the central nervous system, we're looking at a cross-section of the spine. So we're taking information in from receptors. So these could be in the skin, for example, touch receptors, heat receptors, pain receptors. And so we have to transmit that information to the central nervous system. So those neurons that bring information in are sometimes referred to as sensory neurons or afferent neurons. You can see this is a unipolar neuron, and it's transmitting that information into the central nervous system. And then we have information going back out. We're sending information out to effectors in our body. It could be muscles, could be glands. And we're going to call those motor neurons. They're moving away from the center. Sometimes we refer to those as afferent neurons as well. Now, there should be connections between those neurons. Those aren't all the neurons we have in our body. And so we also have these connections in the middle, and those are going to be called interneurons. And so that's the neuron. How did you do? Can you remember the different parts of the neuron? Do you remember that these are the dendrites, the cell body? Do you remember what type of a neuron this is? If not, go back and rewind it. But those are neurons. Those are the basics, and I hope that was.
Skin. And there are other neurons called as interneurons. So these interneurons, the main function of the interneurons is to connect the sensory neuron and motor neurons. Okay. And I'm having another video regarding the synapse. Okay. Okay. What is the main function of synapse? Okay, fine, I will display another video.
Between the input to synapse and the output. But the common types of neuron-neuron relationship is many to one, meaning one neuron takes input from several neurons, creating several synaptic regions. It is very important to know that synaptic connections are mainly on a cell body or in a dendritic membranes, the places where we see ligand-gated channels. We will see how an action potential transmits from one neuron to another. First, let me zoom here a single dendritic synaptic point. The action potential comes and depolarizes the presynaptic membrane. The vesicles release their content into the synaptic cleft. Suppose it releases acetylcholine. This neurotransmitter binds with its receptor and opens channels increasing sodium and potassium conductance. Because the net force on sodium is greater than net force on potassium, the main current flowing is sodium influx. Sodium influx depolarizes the postsynaptic membrane, let's suppose from -70 to 0 millivolt, to a value approximately halfway between the equilibrium potentials for sodium and potassium. It is very important to know that at -10 millivolts, we have the threshold potential. When we reach the threshold potential, the current travels along the dendritic and cell body membranes up to the axon hillock. It is important to know that at dendrites as well as at neuronal body, the current flow cannot initiate an action potential because here we do not have fast voltage-gated sodium channels. The only region near to the dendrites where we do have a high density of voltage-gated sodium channels is an axon hillock. Let me zoom here the membrane of the axon hillock. When a current flows up to the axon hillock, it takes the membrane potential from its resting state up to the threshold potential. As a consequence, the voltage-gated sodium channels open up, and we get a sodium influx, and this sodium influx initiates an action potential. This action potential further transmits forward along the axon. Another important point is that the closer the synapse is to the axon hillock, post-synaptic into one, meaning one neuron takes input from several neurons, creating several synaptic regions. It is very important to know that synaptic connections are mainly on a cell body or in a dendritic membranes, the places where we see ligand-gated channels. We will see how an action potential transmits from one neuron to another. First, let me zoom here a single dendritic synaptic point. The action potential comes and depolarizes the presynaptic membrane. The vesicles release their content into the synaptic cleft. Suppose it releases acetylcholine. This neurotransmitter binds with its receptor and opens channels increasing sodium and potassium conductance. Because the net force on sodium is greater than net force on potassium, the main current flowing is sodium influx. Sodium influx depolarizes the postsynaptic membrane, let's suppose from -70 to 0 millivolt, to a value approximately halfway between the equilibrium potentials for sodium and potassium. It is very important to know that at -10 millivolts, we have the threshold potential. When we reach the threshold potential, the current travels along the dendritic and cell body membranes up to the axon hillock. It is important to know that at dendrites as well as at neuronal body, the current flow cannot initiate an action potential because here we do not have fast voltage-gated sodium channels. The only region near to the dendrites where we do have a high density of voltage-gated sodium channels is an axon hillock. Let me zoom here the membrane of the axon hillock. When a current flows up to the axon hillock, it takes the membrane potential from its resting state out to the threshold potential. As a consequence, the voltage-gated sodium channels open up, and we get a sodium influx, and this sodium influx initiates an action potential. This action potential further transmits forward along the axon. Another important point is that the closer the synapse is to the axon hillock, the greater its influence in determining whether an action potential is generated. In other words, out of these two synapses, the second one has greater chance to generate an action potential because it is closer to the axon hillock. Another important point to understand is that in these synapses, an action potential in a presynaptic cell is insufficient to produce an action potential in a postsynaptic cell. Instead, many presynaptic cells converge on the postsynaptic cell. These inputs summate, and the sum of the inputs determines whether the postsynaptic cell will fire an action potential. In this part of the video, we will talk about the differences between the excitatory postsynaptic potential and inhibitory postsynaptic potential. First, let's talk about excitatory postsynaptic potential. When a presynaptic membrane depolarizes, let's suppose it releases glutamate, an excitatory neurotransmitter. Glutamate attaches to its receptor in a postsynaptic membrane and opens non-selectively permeable channels for sodium and potassium. Because of the net force, sodium influx dominates over potassium efflux. The sodium influx, let's suppose, make the membrane potential less negative, perhaps to negative 60 millivolts. This 10 millivolts of depolarization is called excitatory postsynaptic potential. Excitatory postsynaptic potential brings the membrane potential close to the threshold potential, which is at negative 10 millivolts. This excitatory postsynaptic potential increases the excitability of the postsynaptic neuron, making the neuron more likely to fire an action potential. It is important to note that excitatory postsynaptic potential is similar to the end-plane potential found at the neuromuscular junction and also summates to reach the threshold and generate an action potential. In addition to glutamate, excitatory neurotransmitters include acetylcholine and aspartate.
I think we should explore a few solutions.
Second, let's talk about inhibitory postsynaptic potential. When a presynaptic membrane depolarizes, suppose it releases an inhibitory neurotransmitter, GABA, in a synaptic cleft. GABA attaches to its receptor in a postsynaptic membrane and opens chloride ion channels, a type of ligand-gated channel. Because the membrane potential in a postsynaptic membrane is negative 70 millivolts, and a chloride equilibrium potential is negative 90 millivolts, we get a net force on chloride directed inward. Please note, chloride is negatively charged ion. This chloride influx hyperpolarizes the postsynaptic cell toward its equilibrium potential, suppose from negative 70 to negative 85 millivolts. This 15 millivolts of hyperpolarization is referred to as inhibitory postsynaptic potential. Inhibitory postsynaptic potential takes the membrane potential away from threshold. This decreases the excitability of the postsynaptic neuron, making the neuron further from firing an action potential. In addition of GABA, inhibitory neurotransmitters include glycine. Now let's recap and sum up what we have looked at in this section and add some points which are important for clinical purposes. First, let's talk about decreased neuronal excitability, conduction, and increased neuronal excitability or conduction. Clinical signs of decreased neuronal excitability and conduction could include weakness, ataxia, hyporeflexia, paralysis, and sensory deficit. The causes of decreased neuronal excitability include ion disturbances that include hypokalemia, chronic hyperkalemia, and hypercalcemia. A second possible cause of decreased neuronal excitability and conduction includes loss of neurons, demyelination, as in case of Guillain-Barré, amyotrophic lateral split.
Okay, that's enough for you. I think this video has many informations, and next we are going to start with action potential. Have enough knowledge regarding the synapses and neurons. Okay, wait. Now we can start the other practicals, okay? If you are writing the practicals, we have to start the other practicals separately. Okay, action potential, and this is the most important section. You may have this for your final exams as well. So anyway, I think your 58 students are here. Anyway, I will repeat it if it's possible when you're coming physically. Okay, let's start. Okay, please go through the slide for about 1 minute. Okay. So here action potential is a short lasting event. So it is very shortest event. And here what is happening is the electrical membrane potential of the cell is rapidly rises and falls. Okay. When the neuron is not conducting any impulses, there is some voltage. Okay, that is called as resting potential voltage, and it is about minus 70 millivolts. Okay, that is when neuron is not conducting any impulses. So here during the action potential times, the electrical membrane potential of the cell rapidly rises and falls. Okay, here this action potential is just an event we will study later. This can happen in several type of animal cells, so that are called as excitable cells. So these excitable cells includes neurons, muscle cell, endocrine cells, as well as some plant cells. So these action potentials can happen on neurons, muscle cell, and endocrine cells, as well as some plant cells. Okay. Normally, the main function of neurons is to clear cell-to-cell communications. In other type of cells, the main function is to activate intracellular process. Okay, when considering muscle cells, the main function of action potential is, it is the first step in the chain of events leading to contraction. So in the muscle cells, the function of action potential is to initiate the contractions, but in the neurons, the main function is to have cell-to-cell communication. Okay, the action potential in the neurons can be called as nerves impulses. In other words, it can also called as spikes. Okay, the sequence of action potential is called as spike drain. Okay, again I am repeating, the action potentials in the neuron can be called as nerves impulses or spikes. The sequence of the continuation of this action potential is called as the spike drain. So the neurons which is emitting the action potential can be said that it is in fire. These are just terminologies. Okay. These action potentials are generated by special kind of voltage gated ion channels. Okay, there are sodium ion channels and potassium ion channels are there. And normally, when the neuron is not conducting any signals, the neuron will be at resting potential. It is about minus 70 millivolt. But when this potential increases, so during the resting potentials, these ion channels will be closed. But when the potential increases than a threshold value, these ion channels will open. Okay, these action potentials are generated by some special type of voltage gated ion channels. Okay, these ion channels are in the cells of the plasma membranes. Sorry, cells plasma membranes. And these ion channels will shut down when the membrane potential is near the resting potentials, but the channels will begin to open when the potential increases than the defined threshold level. Okay, please go through this slide. Neurons are on the resting potentials. These ion channels will not open. Okay, when there is a change in the potentials, it is opening. And when the ion channels are opening, they allow the inward flow of sodium ions. Okay, they are allowing the inward flow of sodium ions, and this is changing the electrochemical gradient. So, and it is also producing a further rise in the membrane potential. So when the ion channel is opening, they are allowing the sodium allowed to come inside or inwardly. They are allowing the inward flow of sodium ions, and there is a change in electrochemical gradient. And it is producing a further rise in membrane potentials. So these are the pictures of voltage gated channels. Okay, here the voltage is minus 70 millivolt. That means they are in the resting potentials. So when it is exceeding the threshold level, that is voltage about minus 55 millivolts, so these channels opens and the sodium ions will flow inside. Okay, normally when the when the neuron is at resting potential, sodium ions will be high outside of the cell, and the potassium ions will be high inside the cells. Okay, please remember this. So when the sodium ion channel opens, this causes more channel to open. So it also causes the nearby channels to open, and they will produce a great electric current. Okay. So this process proceeds continuously until all the available ion channels are open, and it will result in a large upswing in the membrane potential. So there is some large changes or upswing in the membrane potentials because of the opening of these ion channels. And this rapid influx of the sodium ion causes the polarity of the plasma membranes to reverse. Okay, so here the depolarization occurring. So this also causing the ion channel to inactivate. Okay, when the sodium channels are closing, the sodium can no longer enter the neurons. So after this, they will be actively transported out of the plasma membranes. So this is the picture of the action potential. So here normally it is the resting potential, that is minus 70 millivolts. So when the sodium ion channel is opening, the influx of the sodium ions will happen, and when it exceeds the threshold levels, so depolarization occurs, and here the potassium channels are opening. That's why it is calling as, so it is causing the repolarizations, and the closing of the potassium channels will take some more time, that is leading to hyperpolarization. The hyperpolarization is responsible to stop the impulse to flow in the backward direction. So here we already studied, so when the sodium ions come inside, it is causing the depolarizations, and at one time the sodium ion channels is closing and the potassium ion channels will be activated. And after the activation of the potassium channels, there is the outward flow of the potassium ions, and they are returning the electrochemical gradient to the resting state. Okay, after action potential has occurred, there is a transient negative shift. Okay, the potassium ion channels are not closing and they are opening for some more time. Because of that, there is some transient negative shift, and that is called as hyperpolarizations, or that is due to additional potassium currents. So this hyperpolarization is responsible for preventing the action potential to traveling back the way it comes. So reverse, it is preventing the reverse flow of that action potentials. Okay. In animal cells, there are two primary type of action potentials. One is generated by voltage-gated sodium channels, and other one is generated by voltage-gated calcium channels. Okay, wait. Potential, okay.
Cells coordinating movement or polarization and repolarization. Hi, it's Paul Anderson, and in this video I'm going to talk about the action potential, that characteristic depolarization and repolarization of an excitable cell. I'm going to be talking about neurons, but we could also have action potentials in muscle cells coordinating movement, or in cells of the endocrine system, for example, coordinating the release of insulin. So if we think about a neuron is getting information from its dendrites and cell body. Remember, they're connected to a lot of other neurons, and based on the summation of all of that information, the neurotransmitters, it's either going to fire an action potential down the axon or not. And so in this video, we're going to think about three different potentials. The resting potential, that's the resting potential of a neuron at rest. It's not really at rest. We then have a graded potential based on the stimulus that that neuron is going to get. And then we may or may not have an action potential. Now that can lead to synaptic activity on the next neuron. I'm going to go through it in the following order. First, we're going to talk about what a resting potential is and how the sodium-potassium pump and the permeability of different ions in the membrane is contributing to the resting potential. We'll then skip forward to the action potential and the importance of voltage-gated channels. And then we'll come back and talk about how chemically gated leak channels contribute to either the firing of an action potential or not. And so what is potential? Potential in physics is simply a separation of charge. And so if we think of a battery, a 9-volt battery having potential, that means that there's a separation of the charge within the battery itself. And it also means that we have potential to do work. And so if we're thinking about a cross-section of a neuron, it also has resting potential. What that means is there's a separation of the charges. Now that separation of the charges is going to be across the membrane, just on the surface of the neuron, but we can measure the voltage. It's negative 70 millivolts in a typical human neuron. And so when you're seeing pictures of an action potential like this, on either side of the action potential, we can see that that neuron is at rest. Now, it doesn't mean that it doesn't require energy to do that. And so let's zoom into the surface of that neuron and see what's going on. And I've got a little mnemonic that I use with my students to remember the important ions on either side of that membrane. I say the neuron is like a salty banana. Most people remember that a banana has a high amount of potassium on the inside. Doesn't mean that it doesn't have potassium on the outside. There's just a higher percent of potassium on the inside. There are also going to be negatively charged anions. For example, proteins on the inside of the neuron are going to have a negative charge. And then since it's a salty banana, on the outside we're going to have salt. We're going to have sodium and chloride ions on the outside as well. They're going to be a higher percentage. We still have them on the inside, but a higher percentage on the outside. Now, why do we have more sodium on the outside, more potassium on the inside? I'll show you in a little bit how the sodium-potassium pump contributes to that. But as you look at this right now, there's still no potential. We don't see a separation of charges. And to really figure out how that works, let's remove a lot of it and just focus on the potassium itself, because it's really the potassium and the permeability of that potassium that's establishing much of that membrane potential. And so we've got a channel on the membrane surface. It's called a leak channel, and it's a leak channel that allows potassium to move out. And so if we think about where is the potassium going to go, there's a chemical gradient from the inside to the outside. In other words, there's a higher concentration of potassium on the inside than on the outside. And since some of it can leak through, it's going to go in that direction. But watch what happens as the potassium moves out. We all of a sudden see a separation in charge. Since this is a positive charge here, it's moving out. We have a separation of that charge, and it's a relatively negative charge on the inside. We also see the arrival of a new gradient. That's the electrical gradient. Since we've got all these positive charges here, they're going to repel each other. They're going to want to move towards the negative charge. And so as more potassium leaves, you can see that electric charge is increasing. And so eventually what could happen is it could start to flow back in. And so we're going to have movement of potassium inside and outside. But we've got an electrochemical gradient outside. Now, why doesn't all the potassium just flow out? It's because these are leak channels. They don't allow much of that potassium to flow out. Now, if we look at a PHET simulation, and I'll put a link to that in the description down below, they've simulated the passing of potassium in and outside of the cell. You can see that we also have another leak channel right here. It's not as busy, but that is a sodium leak channel as well. And so we have an electrochemical gradient of sodium. Remember, there's more sodium on the outside that's coming in. And so we've got these two gradients that are set. You can see the chloride's not moving. You can see that the proteins are not moving as well, and that's because we don't have a channel for them. Now, if we were to just let this go, eventually we would lose that potential because all the sodium would come in. And so we have a sodium-potassium pump where we cash in a little bit of ATP, and that's going to move that sodium out, 3 sodium ions for every 2 potassium ions. And so as this is moving and establishing that membrane potential, again, it's the permeability of these 2 ions that's establishing it. That sodium-potassium pump is essentially keeping up with the sodium that's flowing in and the potassium that's flowing out. And so that is our resting potential. Negative 70 millivolts is established because we have a separation of those ions on the inside and the outside, and their different electrochemical gradients are establishing that difference in the charge. And so we have this resting potential. Now, it requires energy to do that. But what could that resting potential eventually lead to? An action potential. Now, let's look at what an action potential looks like. This is that PHET simulation again. And you can see there's a huge amount of motion between the ions on that surface. And as we zoom in, we see our sodium leak channel, we see our potassium leak channel, but we see another couple of channels that are really, really important. And those are the voltage-gated channels. How do they work? Well, let's look at the sodium voltage-gated channel. So it looks like this. It's got a little polypeptide ball on the bottom. And so when the voltage approaches negative 55 millivolts, it and it opens up wide, and that allows a huge amount of sodium to come into the cell. You can see that rush of sodium in that simulation. Eventually, as we move away from that voltage, it's going to be inactivated, and then it's eventually going to close. There's also potassium voltage-gated channels as well, and they're going to open when the voltage becomes around 30 millivolts. They'll open wide open. What is that going to do? It only allows the passage of potassium. Potassium is going to move outside of the cell. And so as we look at that action potential, once we hit that threshold at negative 55, all of those sodium ion channels, the voltage-gated channels, are going to open wide open. What does that do? It allows a rush of sodium into the cell. What does that do to the voltage? It's going to depolarize that neuron. The voltage is going to increase radically because we have an influx of all of this sodium ions. What eventually happens is we get to 30 millivolts, and those sodium voltage-gated channels are going to become inactivated, and now the potassium channels are going to open wide open. What's going to happen? We have a rush of that potassium outside of the neuron. It takes a while for those to eventually close, and so we then have an undershoot, and what's happened is at this point the voltage has gotten really, really low, but now that permeability, again, those leak channels are going to reestablish that gradient again to negative 70 millivolts, so we could have another action potential. So if we look at what that looks like in slow motion, we've got a quick opening of the sodium voltage-gated channels. You can see an influx of sodium from there and adjacent. That's followed quickly by an opening of the potassium channels. We then added that outflow of that potassium, and then you can see that permeability is going to reestablish us at that resting potential. We could look at what this looks like as we graph the potential over time. Open of sodium, we see a rapid depolarization, opening of the potassium, a repolarization, and then we have that undershoot. And during that time, until we reestablish that gradient at resting potential, we can't have another action potential. Now, so far, we've only been looking at one cross-section of the neuron itself. How does that message move all the way down the axon? Well, if we look at a simulation, it looks like this. Let me slow that down. What really is going on is we're having depolarization of a segment of the axon, which triggers depolarization of the next segment, and the next segment, and the next segment. So let's zoom in and look at those voltage-gated channels and see what's going on. So if we look at, this is near the axon hillock at the beginning of the axon itself. We've got some voltage-gated sodium and potassium channels. And so when we hit that threshold, those sodium channels are going to open wide. We have an influx of sodium ions here, and what they're going to do is they're going to move away from each other since they are going to repel like charges. And they're going to change the voltage in this next segment. What does that do? It hits threshold again and opens up the next sodium channels. Now, why isn't it going to move to the left? Because to the left, we have that repolarization of that segment to the left. So we're opening up those potassium channels, and we have an outflow of those charges. We're not going to be near that threshold for the sodium voltage-gated channels. And so what we're really having on the leading edge of this is a positive feedback loop. We have more sodium coming in, which triggers more opening of those sodium channels. And behind that, we have the undershoot of as we reestablish that gradient again. And so it's going to travel all the way down the axon. Now, this is a simulation of what that electrochemical signal looks like as it moves down. But we have some really long axons in our body. We have an axon that goes all the way from the base of our spinal cord down to our foot. And so it's not going to travel fast enough. And so in a lot of those neurons, they're myelinated. And what that means is they're wrapped with myelin, which insulates that nerve fiber. And so if we look at it like this, what's really going on is we're putting those voltage-gated channels in between that myelin sheath. And I'm not going to get into the specifics of what's going on in here, but if we look at the simulation right here, what it does is allows that electrochemical gradient, you can compare the two, to travel more readily as it moves farther down the neuron. Now, action potentials succumb to what's called the all or none law. And what that means is that an action potential will occur or will not, and it doesn't depend on how much of the stimulus it gets. It depends on if you hit that threshold or not. And so once we hit that threshold, we can move towards it, we can move away from that voltage. But once we hit negative 55 millivolts, we're going to have an action potential, and that action potential is going to look exactly the same. Now, in this diagram right here, we have the three different potentials that I'm talking about in this video. At the beginning, we have the resting potential. Remember, that's contributed to by the permeability of the membrane to different ions. We then have the action potential, that rapid depolarization and repolarization of the neuron. What's going on in here, we have graded potentials. In other words, the potential is moving towards or away from that threshold. And so if we think about what's going on there, to really understand that, we're getting information from other nerves. There are synapses here, and those synapses are releasing neurotransmitters. Those neurotransmitters are moving across the synapse, and they're opening up a new form of channel that's important when we talk about graded potentials. Those are chemically graded channels. And so these neurotransmitters are going to dock with proteins on the postsynaptic side of those neurons, and what they'll do is they'll open it up. What does that do? Well, it depends on what type of neurotransmitter it is and what type of receptor it is. There are some that are going to be excitatory. And if we look back at our graded potentials, excitatory means that they're moving us towards the threshold, towards that all or none action potential to occur. What's the easiest way to do that? If we open up the channel and we have a channel that allows sodium to run in, that's going to depolarize us and it's going to move us to that threshold, just like those sodium-gated channels are going to do. And we also have inhibitory neurotransmitters and receptors. What are they going to do? They're going to move away from that threshold. What's the easiest way to do that is to have a receptor that's going to allow potassium to rush out, or you could allow fluoride to come into that neuron itself, and it's going to push us away from that threshold. So we can think of a neuron as an analog portion and a digital portion. So since it's all or none down here, we're either going to have the action potential or not. So it's like a 1 or a 0. But if we look on this side of the neuron, where the cell body is and the dendrites are, we're getting a lot of information from neurotransmitters of neurons that are attached to the dendrites and the cell body of this neuron itself. Some of those are excitatory. What they're doing is depolarizing that neuron. They're increasing the voltage, pushing us closer to that threshold. And some of those are going to be inhibitory. They're pushing us away from it. And then, so it's the summation of all of those signals that's telling that neuron to either fire an action potential if we hit negative 55 or not. And so that is the all or none law. And so in summary, what is the resting potential? It's negative 70 millivolts. It's where a neuron is sitting. It has a potential to do work. It's established by the permeability of the membranes and the sodium-potassium pump. We then moved on to the graded potential, and that's based on all the neurotransmitters that we're getting from connected neurons. And then if we hit that negative 55, we're going to have an opening of those sodium ion channels, and we're going to have an all or none action potential that runs down to the neuron. So that's the action potential, and I hope that was it.
I think with that you can get a knowledge from what is action potential, okay? So with that we can end up the class and we can conduct the next portion of the action potential in the coming classes, okay?